So I am visible, audible, everything all right? Visibility is there, audibility is there, right? Good morning, good morning, good morning. Okay, I will ask the admin to send the exam paper. Probably paper are also in your app, na? So just have a look. So today we are starting with Golgi complex. We are starting with Golgi complex. Okay, we will discuss about that question. Which question you are finding you have doubt about the test? What question was there? We will talk later, okay? Keep your question with you. So, start with the Golgi complex. So, if we talk about Golgi complex, it is discovered by C. Golgi, Camillo Golgi. C. Golgi discovered it. Got the idea? What they observe, they find as densely stained reticular structure near the nucleus. Near the nucleus. So it is discovered by Camillo Golgi as densely stained reticular structure near the nucleus. Later on, called Golgi body or Golgi complex. Complex after him. Because it is discovered by Golgi, na? So in honor of his discoverer, we just gave the name as what? Golgi complex. Got the idea? And that is the reason your textbook wrote. It is later on called Golgi complex after him. It is also called lipochondria. Golgi body is also called what? Lipochondria. Lipochondria, that is meaning is what? Fat bodies. In plant, Golgi complex is called dictyosome. It is called dictyosome in plant. It is called dictyosome in plant. So Golgi complex has some another name also. It is also called lipochondria and it is called dictyosome. But we use the word dictyosome in case of plant. Got the idea? Is it clear? Now we are going to talk about parts of Golgi body. First component is called cisternae. Second component is called tubule, and the third component is vesicle. So these are the part or component of Golgi complex. You can also write component. Component or part of Golgi complex. How many part I told? 3 parts. Cisternae, tubules and vesicle. Right? Now we are moving ahead. So if we talk about cisternae, it is somehow appear like this. It is somehow concentrically arranged So I am just telling you with the diagram of you will got better. Suppose that this is a cell. And these structures are what basically cisternae. So these cisternae are even interconnected. Got the idea? This is cisternae. Suppose that for your understanding I am telling it is nucleus. It is nucleus. Got the idea? So what you are finding, you are just finding somehow concentrically arranged flattened structure which is connected to each other, arranged with a polarity. Toward the nucleus, you are finding what is there? Convex or concave phase? Convex phase. And toward the cell membrane, it is concave phase. Got the idea? Toward the nucleus, it is arranging like a concentrically, sinking na, it is looking like a concentric structure, looking like a ring-like structure it is forming. So in a ring, what you are finding, a convex face is there and a concave face is there. Concave face is facing the plasma membrane and convex face facing the nucleus. And this is what basically convex face, this is called concave face. This convex face is also called cis face, or it is also called forming face. Forming face, other name of the convex face of the cisternae. If we talk about concave face, it is also called transface, or it is also called maturing phase. Maturing phase. Got the idea? So concave have a other name, transphase or maturing phase. So if we talk about cisterna, you got the idea? If we talk about the diameter of the tubules, cisternal tubule is how much? 0.5 to 1 micrometer. This is the diameter. Got the idea? And number of cisterna is variable from Golgi body to Golgi body. Number of cisternae is variable. Cisternae are interconnected structure. Interconnected structure. Did you got the idea? These are interconnected structure. So one component you got. Now another component is tubule. So if I draw the structure of tubules of the Golgi complex, you can see like this. It is somehow tubular structure, branch, irregular, tubular structure. I am just making it like what? Tubule. It is branched, irregular, tubular structure. And another one is vesicle. All three components I made here. Vesicle. So these are the structure overall, all three overall called which one? Golgi complex. Got the idea? So all the structures, cisternae, tubule, vesicle, all together making what? Basically they all are arranged together and they are called what? Basically Golgi complex. And these are the three components of the Golgi complex. This yellow color here is what? Cisternae, this one is tubule, this one is what? Basically vesicles. Got the idea? A structure of Golgi complex came in mind. Right? It is toward side of the nucleus. It is not connected with nucleus. Convex face or cis face or forming face is toward nucleus, not connected with nucleus, toward nucleus. And concave face, trans face or maturing face is toward plasma membrane. Got the idea? So Golgi complex is a cell organelle with polarity. Two poles are there, cis and trans, convex and concave. Golgi complex is a cell organelle with polarity. Got the idea? Golgi complex is a cell organelle with polarity. So what I told till now, let me recap, then I will move ahead once again. So let me recap since beginning. Then you will got a better connectivity. So let's start since beginning. Haji. So let me revise it once, recap it once. So look here, I am starting with Golgi complex. Just give your attention now, you will understand everything. So Golgi complex is discovered by Camillo Golgi. Who discovered it? Camillo Golgi discovered it. Got the idea? How they discovered it? They find there is a densely stained reticular structure present near the nucleus. Later on, this densely stained reticular structure who are present near the nucleus is called Golgi complex in the honor of Camillo Golgi, who is the discoverer of it. So I hope up to here it is all right. Any problem till here? No problem, I hope. Got the idea? Is it clear? Done? Now I am coming on next. I am telling what are the other names of the Golgi complex. So I told about other name of Golgi complex. It is also called lipochondria. What is the meaning of lipochondria? It is called fat bodies. Lipochondria is called fat bodies. Getting the things or not? Or It is also called dictyosome in plant. The word dictyosome we use in the case of which one? Plant. Got the idea? Lipochondria or plant. Now, what are the component of Golgi complex? So I wrote here 3 different components are present in Golgi complex. One is cisternae, another is tubules, another is vesicle. So in this diagram, these 3 components are there. These concentric, see na, it is looking like a ring, part of ring. It is a ring. Just a minute, I am just making a ring. Have a look. I am making a ring. Got the idea? Ring within a ring. I am making like. And I am just showing you that part of ring only. So it is looking like a concentrically arranged structure. It is looking like which one? A concentrically arranged structure. Are you getting the idea? That is the reason I am writing the word concentric. Concentric means what? Looking like a ring within a ring. It is a part of a ring. It is looking like part of a ring. So it is concentrically arranged structure. Got the idea? Concentrically arranged flattened structure. Or if you talk these cisternae, here one, here first I am writing, here second, here third, here fourth, here five. So what I wrote, these ring like a structure is variable from Golgi complex to Golgi complex and they are interconnected. If we talk about the diameter of this cisternae, I wrote about the diameter of this cisternae as what? 0.5 to 1 micron. So I told you about the cisternae. It is concentrically arranged, flattened structure. Number is variable. It is not fixed in a particular cell. It is variable from cell type to cell type. Another thing, it is interconnected structure. Got the idea? So it is called cis phase. Who are convex phase is called cis phase, and who are concave phase is called trans phase. So here poles are forming. One pole is called convex pole, another pole is called concave pole. It is called cis pole, it is called trans pole. So they are made up of same material, but they are entirely different. Cis and trans phase are entirely different. Beside of cisternae, who is present? Tube-like irregular branch structure is present, that is called tubule. And beside of that, some oval or a spherical structure is also present, that is called vesicle. So all 3 together are called what? Basically Golgi complex. Up to here, any problem? Got the idea? Clear? Now we are moving ahead. Now we are going to talk about function of Golgi complex. A structure we discussed already. Function of Golgi complex. So functions of the Golgi complex is, first function writing packaging. Second function I am writing secretion. Third function I am writing chemical modification. Got idea? What in chemical modification? That is for glycosylation or glycosidation. Glycosylation, that is formation of glycoprotein and glycolipid. Another function I am writing formation of lysosome. Next function I am writing have role in formation of phragmoplast and cell wall in plant. Got the idea? So these are the some of the functions of Golgi complex. Now I am telling about how the things are going on during process of chemical modification. How these chemical modification occur? Got the idea? How the chemical modification occur? Got the idea? Another things you can say about function, transport of macromolecule to intracellular target or outside of cell. So these are the functions of the Golgi complex. Once again, see about the function what I wrote. First function I wrote packaging. Second function I wrote secretion. Third function I wrote chemical modification. What is meaning of chemical modification? Glycosylation. What is glycosylation? Formation of glycoprotein and glycolipid is called glycosylation. Other function is formation of lysosome. Another function is formations of phragmoplast or cell wall. It not only play a role, it is a part who are playing a role in their formation. And transport of macromolecule to intracellular target or outside of the cell. So Golgi body is also called, it is also called middleman of the cell. Middleman of the cell. If you talk about, it is also called director of macromolecular traffic. Why it is called, I am going to explain. But before that, I am just going to say about how it is going for chemical modification, glycosylation. Got the idea? That is formation of glycoprotein and glycolipid. So these are the functions, keep in your mind. Is it clear? Now I am going to explain about glycosylation. During cell wall, I use a word na phragmoplast. Vesicle of during many question coming for what is phragmoplast. I am telling you first what is phragmoplast. Many people are asking, commenting what is phragmoplast. Suppose that it is a plant cell and a plant cell divided here into two cells. Sorry, one nuclei divided into two nuclei. Now cell wall formation occur. So the vesicle of Golgi complex arranged like this. Not only vesicle of Golgi complex is there, many other things are there also. That is the reason one of the part is what? Vesicle of Golgi complex. So these vesicle of Golgi complex, here microfilament is also there. Endoplasmic reticulum also play a role. And this structure who is formed is called phragmoplast. These phragmoplasts later converted into which one? Middle lamella, sorry, cell plate. And then cell plate later converted into which one? Middle lamella. Did you got the idea? So these phragmoplasts formation, in this phragmoplast formation, who play a role? Golgi complex play a role. It is not exclusively making it, but it is a part who are playing a role in the formation of this phragmoplast during cell division in a plant. I will tell you during when we will teach about cell division, how cell plate formation occur in the plant cell. Not, I told in the beginning, that there is a right place to deal everything. So this is not the right time to talk in much more detail about the phragmoplast. Okay. So now give your attention. So I am just telling you about how chemical modification occurs. Just try to understand this is a cell and it is important, give your attention. This is a cell, this is the nucleus. I'm just giving a symbolic diagram. Now I'm just telling, suppose that this is a cell organelle, you can say mitochondria. An example, suppose that this is endoplasmic reticulum. Suppose that it is endoplasmic reticulum. Here ribosome is there, so it is what? Rough endoplasmic reticulum. Here RER, and here ribosome is not there, so it is SER. This is ER. Now I am making Golgi complex. Just try to connect. Suppose that this is Golgi complex. NCERT wrote, Golgi complex is present near the ER. This is the cisternae I am making. This is a cell organelle which is present near the endoplasmic reticulum. So this is which phase? Cis phase. And this is which one? Trans phase. This is what basically trans phase. Now just try to connect how Golgi complex working. And here you will understand one more thing. How cell organelle undergoes with a coordinated function. I told both Golgi complex, both Golgi complex, ER, or even lysosome and vacuole all together forming a endomembranous system. Got the idea? I told example of endomembranous system, Golgi complex, ER, which one? Vacuoles and lysosome, these four cell organelle I told making a endomembranous system because their functions are coordinated. Because their functions are coordinated. Now just give your attention what happening here. This is ribosome. So ribosome is a cell organelle. What is its function? One-liner function I am writing. Function of ribosome is protein synthesis. Function of ribosome is protein synthesis. So protein synthesis occur on ribosome. What I am writing? I am writing chemical modification steps. What happening? Chemical modification I am writing stepwise. You will understand better. Modification. So very first job what happened? Protein, I am telling in reference of protein first. Protein synthesis occur on ribosome which attached with endoplasmic reticulum. ER not do protein synthesis. Ribosome attached with ER do the protein synthesis. Are you getting the differences or not? Many times students confuse. ER do the protein synthesis. Getting the things? ER not do the protein synthesis. Who is doing the protein synthesis? I told earlier it is ER. and it is ribosome. Ribosome doing the protein synthesis. Getting the idea? Later on, what the second step occur? In second step, protein enter inside the lumen of ER. Got the idea? Protein enter inside the lumen of ER. What happened inside the lumen of ER? Inside of lumen of ER, protein undergoes in structural changes. A structural modification occur in protein. Actually, when new protein form, it is like this. It is amino acid because many amino acid are present in a protein. So many amino acid, I will tell you in the latter, you will not understand much here if I say, this is called primary structure. Primary structure of the protein looking like a thread of wool. So it is a thread like a structure. When it is folding like this, it becomes secondary structure. Many other type of secondary structure is there, but I am not telling right now everything here. There is a respective chapter where I will deal about it, secondary structure. When protein become like this, it is called tertiary structure. Many fold occur. And when more than one tertiary is joined like this, it is making a quaternary structure. So there are different structure of protein. So when a new protein form on a ribosome, it present in this form. So from this form to this form or this form or this form, these all changes of the protein structure occur inside the lumen of the ER. You got the basic idea? You got the basic idea? Is it? You got the basic idea? Yes. So this structure, primary structure converted into secondary, secondary to tertiary, tertiary to quaternary structure. These all changes occur in the which one? Endoplasmic reticulum. Now what happened? After these structural change, I am writing note. Chemical modification may occur in ER also. But main area of chemical modification is what? Golgi complex. May occur in ER also. ER also. But mainly occur in Golgi complex. Mainly occur in Golgi complex. Got the idea? Now let me show you through the diagram. Now here is the protein. Here is the protein. Suppose that it is undergoes in a structural modification. Now what happened? These protein who synthesize on ribosome packed in vesicle of ER. So these are going to pinch off and making a vesicle. Who is going to pinch off? Getting the things. And here it is a vesicle. In vesicle, who is packed? It is vesicle of ER. Vesicle also present in ER, vesicle also present in Golgi complex. So this is vesicle of ER. Now these vesicle going to fuse with cis phase. Here it is fusing with which one? Cis phase. Fuse with cis phase. And as it fuse with cis phase, they release the protein in the cisternae near the cis phase. Now, because you better know, cisterna are interconnected, so these protein moving via cis phase to trans phase. What the idea? These protein moving via cis phase to trans phase. So, and during it is moving via cis phase to trans phase, during this time chemical modification occur. During this time chemical modification occur. So let me write first. So up to here I wrote, I hope you understand. I am just writing in sequence so you will understand better. Very first I wrote protein synthesis occur on ribosome which attach with ER. I hope it is okay. Then protein enter inside the lumen of ER. From outside it enter inside of the lumen. Suppose that here protein synthesis occur outside and then protein on ribosome and then it enter inside. So protein enter in the lumen of inside, it is okay. Inside of lumen protein undergoes in folding. Protein undergoes in folding. So inside the lumen lumen of ER, protein undergoes structural changes. What is these structural changes? Folding. Folding occur. Primary structure convert into secondary, tertiary or quaternary structure. Later on what happened? Later on, fourth step I am writing. Protein now packed in vesicle of ER. Vesicle of ER and fused with cis phase of Golgi complex. Fused with cis phase of the Golgi complex. Let me write, then I will explain. Now what happened? Fifth stage. Protein transported from cis phase to trans phase. Cis phase to trans phase. During that time, chemical modification occur. Chemical modification occur. That is formation of glycoprotein or you can say, if it is lipid also synthesized in Golgi complex, na glycolipid. Sorry, lipid also synthesized in ER. So it is, if it is lipid, it is going to form glycolipid. Lipid also synthesized in which one? ER. So glycoprotein is forming or glycolipid is forming. If it is lipid coming packed in the vesicle, during chemical modification it convert into glycolipid. If protein packed in the vesicle, during chemical modification it is converted into glycoprotein. Now what happened? Chemically modified protein now packed in Golgian vesicle. Golgian vesicle. And these Golgian vesicle transport chemically modified macromolecule to intracellular target or extracellular target. Extracellular target. Got the idea? So I noted it, the entire sequence of event. Now I am just going to say you about once again through the diagram how it's happened. So you can write fifth, you can write it sixth, you can write it seventh. Got the idea? So these are the different event occur during chemical modification. Did you got the idea? Lipid also synthesize in where the lipid and glycolipid synthesis occur. Lipid synthesis occur in which one? Endoplasmic reticulum. Or which endoplasmic reticulum? Smooth endoplasmic reticulum. Lipid and steroidal hormone synthesis occur in a smooth endoplasmic reticulum. I told na. Yeah, I am explaining once again. Don't worry. I just wrote it out. Then I am explaining it. Just a minute. Just a minute. Now see, it is not difficult, it is easy. You just try to connect yourself. I just came on this diagram. I am amplifying this diagram only. We have a look here. First, protein synthesis occur on ribosome. Assume it is the protein. Assume it is the protein. Protein synthesis occur on which one? Ribosome. Ribosome present on ER, so it is making which ER? Rough ER. Now this protein enter inside the lumen. Protein enter inside the lumen. Clear? Now the protein who enter inside the lumen undergoes in a structural changes. Undergoes in a structural changes. A structural changes means what? Primary structure convert into secondary structure. Protein when formed up like this. When it is going for structural changes, it become like this. Later it become like this. So these are primary structure, secondary structure, tertiary structure. Are you getting the idea? So these structural changes occur in the protein. Later on, this protein going to come out from this lumen and packed in a vesicle of ER. This is called vesicle of ER. Now this vesicle of ER fuse with cis phase of the Golgi complex. Fuse with cis phase of the Golgi complex and release their protein inside the cisternae. Now what happened to this protein? This protein going toward transphase. From cis phase, who is traveling toward transphase? Protein traveling toward transphase. And when this protein traveling toward transphase, in that time who occur? Chemical modification occur. Chemical modification occur. Glycosylation occur. Carbohydrate attach with it. And it is going to convert into glycoprotein. I am telling in reference of protein. So normal protein get converted into glycoprotein during this transport from cis phase to transphase. Now this chemically modified protein, what happened? This cisternae is going to pinch off. Pinch off. Pinch off means I am just, you think like it is the membrane of cisternae and something fill here. I am cutting it and removing it. So it is going to pinch off, and here who is present? Glycoprotein is present. So these are the vesicle where glycoprotein is packed. Where glycoprotein is packed. So different chemicals are packed in this vesicle. Different chemicals are packed in this vesicle, and that is the reason we are telling one of the function of the Golgi complex is what? Packaging. They are packing different chemical in this different vesicle. Now this vesicle, what happened? If fuse with, so many such vesicle are there. I am making many such vesicle where chemically modified macromolecules are there. It may be glycoprotein, it may be glycolipid. One I told na, as one forming. So some of these packed vesicle fuse with the cell organelle. It is called intracellular target. This is mitochondria where it transport the macromolecule to intracellular target. If it fuse with the cell membrane, it is releasing these macromolecule outside. It is called secretion. And it is called transport of macromolecule to intracellular target, like mitochondria, another cell organelle. So when it is transporting the chemical to intracellular target, got the idea? And either it is transporting the molecule to outside of the cell. When it is transporting outside the cell, it is called secretion. No any chemical can go anywhere without the permission of Golgi complex, because how they are going? It is going through vesicle, and that is the reason we are telling it middleman of the cell. It is doing where the molecule will go. It is also called director of macromolecular traffic. You got the idea? Understanding came in the mind or not came? Or should I give a more revision? Because my teaching is not important, your understanding is important. So I will say as long as you not understand. Understood or not understood? Say honestly. So most of the student writing, understand, someone writing, time is there still. Class is up to 10:10, not up to 10 o'clock. So listen it carefully. I am just explaining once again. 1 minute it will take. 1 minute it will take. Listen it step by step. Protein synthesis occur on, I will say, you write. Protein synthesis occur on ribosome. This is synthesized protein. Then this protein enter in lumen of ER. Inside the lumen of ER, protein undergoes in structural changes. Primary to secondary, secondary to tertiary structure formed. Got the idea? After structural changes, these protein packed in a vesicle of ER. Later on, this vesicle of ER fuse with cis phase of the Golgi complex and release their protein in the cisternae. Now this protein migrating from cis phase to trans phase. During that time, chemical modification occurs. At trans phase, the cisternal part pinch off and it is forming a packaging material called what? Basically vesicle. So these chemically modified protein packed in the vesicle, and these vesicle may transport this chemically modified protein either to intracellular target or extracellular target. So vesicle is packing this macromolecule. So one of the function of Golgi complex is packaging. It is releasing this material outside of the cell and it is called secretion, and it may transport this material to the intracellular target. Got the idea? Is it clear in the mind? And that I wrote in the language. The protein synthesis occur on ribosome, which is attached to ER. Protein enter inside the lumen of ER. Inside the lumen of ER, what happened? Protein undergoes in structural changes, means folding occur. Due to folding, primary convert into secondary, secondary convert into tertiary, tertiary convert into quaternary structure. Got the idea? Later on, I wrote protein now packed in the vesicle of ER and fused with the cis phase of the Golgi complex. Later on, what happened? Protein transported from cis phase to trans phase. During that time, chemical modification occur and formation of what occur? Glycoprotein occur. If lipid produced in the smooth endoplasmic reticulum, it packed in the vesicle and then it convert into glycolipid. If protein is there, glycoprotein. If lipid is there, glycolipid. These are called chemical modification. Now chemically modified protein now packed in Golgi vesicle, sac-like structure. And these vesicle transport this protein either to what basically? Intracellular target or extracellular target. Getting the things or not? Extracellular means what? Outside of cell. And when it is transporting the things outside of the cell, it is called secretion. Got the idea? Extracellular target by which process? Secretion process. So I hope you understand about this coordinated function of ER and Golgi complex. Coordinated function of ER and Golgi complex. Is it clear? Is it all right? Understood? So how many function I wrote for Golgi complex? Do you know? I wrote these function. Where is that slide? Let me come on that slide. Wait a minute. Here I wrote the function. Don't forget it. I wrote the function of Golgi complex what? Packaging. Packaging means here it is vesicle and packing the material here. Secretion, when they fuse with the cell membrane, they came out, secreted out of the cell. Chemical modification, you know now chemical modification. Formation of lysosome, in next class I will tell how they form the lysosome. In next class I will tell the lysosome, no? There I will tell how it form the lysosome. Now I wrote, it have a role in formation of phragmoplast and cell wall in plant. Got the idea? Transport of macromolecule to intracellular target or outside of the cell, I already explained. And it is also called what? Middleman of the cell. Because any molecule going outside the cell with the help of which one? Golgi complex. Got the idea? And it is also called director of macromolecular traffic. Because any molecule cannot go out of the cell without the permission of Golgi complex. Got the idea? Is it? So one student writing, I have doubt about the test. I hope one question is wrong. Which one is that one question? Please tell me. Let me read if I can. It is correct, dear. Quasi-fluid nature enable the movement of protein in the lipid bilayer. Quasi-fluid nature of lipid enable the movement of protein. I explained that question also. Got the idea? So this is the QR code. Sir, this class related of which class I might have missed? We are dealing cell, the unit of life, and we are dealing Golgi complex. But I told much more than NCERT. Because I just want to explain you what? Chemical modification, how it occur. Got the idea? Right. Okay, so you discuss when you come in that campus. I am not able to connect you better. Quasi-fluid nature of lipid allow lateral movement of protein. That is the answer. So you do the answer like this. Quasi-fluid nature of lipid allow the movement of protein. Okay, done. Bye-bye. Have a great day to all of you. Thank you very much. Write the gift exam. Not a issue, na? You write it. Then you will find how much you are understanding. Bye-bye, bye-bye, bye-bye. Have a great day to all of you. Thank you, children. Bye. See you in the next class. Thank you, thank you, thank you. Bye-bye.